Hi, I'm Isha Jain. I'm a core investigator at the ARC Institute and an associate professor at the Gladstone Institutes and UCSF. My lab is really interested in how organisms interact with the outside world, and more specifically, how what we breathe and what we eat affect our metabolism and physiology. Within that, we're focused on how molecular oxygen in the air and the vitamins in our diet affect disease progression, and how we can use this information to create creative new therapies. Today, I'll tell you about a very exciting new project in the lab about using vitamin B3 as a therapy for NAXD disease. As I mentioned, our health is really dictated by interactions between our genes and our external environment. Within that, diet is extremely important. And within the diet category, micronutrients such as vitamins are absolutely essential for our health. We like to be inspired by the past, and actually this project started when we were reading about the history of how vitamins were discovered. It turns out that there were the so-called vitamin hunters of the late 1800s and early 1900s. At that point, we actually didn't even know what a vitamin was. And it was only observations such as those made by Christian Eichmann that discovered their presence. He noticed that in a group of chickens on a farm, all of a sudden they became very, very sick without any reason that could be explained. And he noticed that this coincided with a change in their diet from unpolished rice to polished rice. And this led to the really unique and exciting discovery that there was something in the rice husks that was essential for life, and ultimately led to the isolation of thiamine, which is vitamin B1. And so because it was a vital amine, vita-amine, hence the term vitamins came about. And this simple discovery kick-started an entire era of vitamin hunters, where over 50 years, about 13 vitamins were discovered. And over a dozen Nobel Prizes were awarded for the discovery of these micronutrients. So what is a vitamin? It's a nutrient that's essential for life, but not made in sufficient quantities within the human body. We like to joke that now we live in the sort of whole foods and supplements era, where people are consuming vitamins in a very ad hoc manner at different doses, different time frequencies, and it's unclear in which conditions high doses of vitamins can actually help. And so we decided to revisit this age-old question of which diseases are likely to benefit from each vitamin and why, thinking that we could have a dramatic impact on human health by understanding the interactions between vitamins and genes. Before I start, I would like to introduce you to the 21st century vitamin hunters in our lab. And so this work was really done by Ankur Garg and Skylar Bloom, who are the superstars that really toiled away to all the hard work to make this discovery happen. We like to say that they're revitalizing this ancient field of vitamin biology using modern methodologies like genomics and CRISPR. And the reason that we're so excited about vitamins as therapies is because they're actually very simple to administer, they're cheap, they're well established from a safety perspective, and there's no regulatory hurdles. Patients can go to the grocery store and get the vitamins the next day. And so we've proposed this new framework thinking about this concept, where instead of starting with a single disease and trying to develop a therapy, instead we're starting with a therapy, in this case a specific vitamin, and asking what are all genetic diseases that are theoretically treatable by that given vitamin. And so to do this, we can do one big experiment where we perturb every single gene in the genome using CRISPR, and ask, out of all the genes in the genome, which ones have a correspondence to a human disease, such that if that gene is broken or mutated, it causes a disease, and it can be rescued by a specific vitamin in our experiment. We first applied this to vitamins B2 and B3, and today I'll tell you mostly about our work on vitamin B3. So vitamin B3 is niacin and is absolutely essential for life. So when we did this large experiment, we nominated dozens of disease genes that could be rescued or treated by vitamin B3 in isolated cells. So it was one big experiment, and it prioritized different diseases for this approach. As a first proof of concept, we took the top hit in this experiment, which was the gene called NAXD. It turns out when you have mutations in this essential gene, it causes a very severe disease in children, and these individuals have severe neurodevelopmental delay, skin lesions, and they die within the first few months or sooner of life. And there aren't well-established treatments that have been shown to work throughout this patient population. And so we decided to ask, can vitamin B3 actually affect this disease? It turns out that this is actually a fairly complicated pathway, and so we first asked what goes wrong in this type of a disease, building on beautiful work by Carol Linster and others in the field. We showed that there is a broken version of vitamin B3 that accumulates in our tissues, and the gene NAXD makes a protein that cleans up this broken version of the vitamin. And so what we found was that simply giving the good version of the vitamin overcomes this accumulation of the broken version and helps in this disease condition. To test whether this was relevant in an animal and not just in isolated cells, we developed the first mouse model of NAXD deficiency, where that gene was deleted in mice. And excitingly, we showed that this model actually very accurately recapitulates the human disease in terms of the brain lesions and the skin lesions, and also the biochemical defects. And so then the big question was, can we treat this model with vitamin B3? So it turns out this mouse model that we developed dies at about 5 days of birth if placed on a normal diet. So very severe disease, similar to the human patients. Very excitingly, we found that simply giving very high doses of vitamin B3 after birth seemed to essentially cure this mouse model, in that giving it daily resulted in over a 40-fold lifespan extension. And so these animals were still alive at a year and probably would have continued to live if we had kept the experiment going. So a very, very dramatic effect size just by giving a daily dose of a vitamin. And this is super exciting because these patients can go to the grocery store tomorrow and start to take higher levels of vitamin B3. And there's actually already clues and case reports suggesting that this might actually be a viable approach. And so we're very excited about the potential that this has for this disease. We then went a little bit deeper and asked what were the cell types that were damaged in the brains of this disease model. And we found that it was a specific cell type, endothelial cells, which line the blood vessels of the brain, that seemed particularly affected in this disease and completely rescued by vitamin B3, suggesting that different cells in our body might have different susceptibilities to these broken metabolites. And by understanding this, we can even refine our therapeutic approach more so. We're excited about this, not only for NAXD disease, but because we think this really opens up the avenues and the doors for a broader concept of precision nutrition. In this context, we really look forward to a future where every single neonate or newborn will be sequenced at birth, and then based on their genetic mutations, either mild mutations that we all have or more severe mutations, they can be prescribed a custom vitamin or micronutrient cocktail that they take throughout their lives. And so instead of having a recommended dietary or daily allowance for vitamins, we'll have a required genetic allowance and really optimize our health based on our genes. So to summarize, we have identified dozens of diseases that might be treatable by vitamins B2 and B3. We now understand a bit more about how the broken version of vitamin B3 contributes to disease. We see this very dramatic lifespan extension just by giving vitamin B3 in the very severe disease model that we generated. And we think that this really opens up the doors for a broader concept or next phase of nutrigenomics. And we hope that other labs will join us in applying this to other micronutrients. With that, a huge thank you to the Jain Lab members. This would not have been possible without all of their hard work. And thank you for listening.